Hello, everybody. Welcome to this virtual event. And I am Emily Leproux, the CEO and co-founder of Twist Bioscience. I'm very excited to talk to you about how Twist is helping writing the future. I'll start by saying that I will be making some forward-looking statement today since we're a public company. So at Twist, we're very excited because DNA is changing the world for the better, and our customers are using DNA to do great things. For instance, if we look on the left here, it's wild teosinte, which has been turned into corn by our ancestors, but that took thousands of years. And now our customers can do the same thing in the laboratory much quicker. Our customers also can, for instance, modify bacteria, engineer bacteria to deliver fertilizer at the root of plants, and that eliminates the need for fertilizer, which is much better from a sustainability point of view, and it also reduces the cost of food. Our customers are also engineering E. coli and to enable the production of chemicals through fermentation. And so we all know fermentation. You can transform sugar into a number of chemicals, so alcohol, vinegar. But if you engineer the genes of E. coli or yeast, you can produce any chemicals you want, and you can't tell the difference. It's the exact same chemical formula. So again, the idea is to make production of chemicals more sustainable and cheaper. In the area of health, our customers are modifying cells or discovering antibodies to be used as therapies. And the power of DNA is such that in the not so distant future, cancer may be a chronic disease. And then last but not least, you can even use DNA to store data for the long term. I don't know if you know, but in California, 3% of the energy used in California is used for data centers, to cool data centers. And so with DNA, there's a potential to, again, have storage of data that is more sustainable and cheaper. So we've done that at Twist by using our silicon platform to write DNA. And on silicon, what we've been able to do is to miniaturize the chemistry of writing DNA. That chemistry is known. And through miniaturization, we can enable two things. One is massive throughput. We can make massive number of oligos and a much lower cost of DNA. And that enables you to do things that you could not do before. And oligos are important because to make genes, for instance, this publication from 1995 is the first gene synthesis publication. Pim Stemmer and his team started from, in this case, a bit more than 50 oligos. And with the right sequence, the oligos hybridize to each other, and with the right enzyme, the 9 kb insert was produced on the left. And on the right, with 130 oligos, again with the right sequence and the right enzyme, they were able to produce a 2.7 kb insert. So if you can make oligos, you're able to assemble those oligos into genes or plasmid. And so at Twist, we've taken the known chemistry of making oligos, we've put it on silicon, and we've reduced the cost and increased the throughput. And thanks to that, we now offer a number of products that enable our customers to really harness the power of DNA for the better. So in the next few slides, I'm not really going to talk about the products. More importantly, I think I'll talk about the application, what our customers do with those tools, and leverage the power of DNA for the better. So first, talking about COVID, it's hard to escape COVID. It was such a disruption to the world, and so at Twist we've used our DNA to help in the response of the pandemic. And there's three main areas we worked on. First is with NGS panels. And so we've built panels where we can sequence the virus in a patient to, one, detect is there a virus, yes or no, and second, being able to read the entire sequence of the virus in order to be able to tell the medical profession which mutant is present. And I'm very glad to report that in the last few weeks we received FDA authorization to use this panel as a diagnostic kit. We also have built synthetic control for companies that are making tests for COVID, or for labs that are running day-to-day COVID tests. It's important to have a control where you know that you have COVID in this tube. But making a positive control is dangerous. Growing the virus would be dangerous. And so we made COVID in a tube in a way that's safe. So it's the full RNA sequence, but cut in 5 pieces. So it's safe, and it's been very useful to people fighting the virus. And then last, we've also developed our own antibodies against COVID. We've done it very quickly, and I'm glad to report that the data is actually really good in an hamster model. You can see here that even in black, at 1 milligrams per kilogram, you have very potent neutralization of the virus from multiple twist antibodies. So we're very glad that we've been able to contribute to the fight against COVID. So maybe moving on to more generally human health, we believe that DNA and our tools are enabling of progress in making sure that human health gets better over time. That's very important. And so maybe first I'll start with target identification. As you know, the first step of doing drug discovery is to know which target should I go after. And our tools, our ability to make oligos have been used by researchers because we can make a guide RNA and then we can make a pool of guide RNA. And then that pool can be used to compare before or after treatment and be able to know which part of the genome is important for that phenotype. And for instance, here on the right, there is a publication from the Sanger Institute in the UK where they use the twist oligo pools on 324 cancer lines. And they were able to find important regions of the genome for those cancer lines. So you can link phenotype to genotype by using CRISPR screening genome-wide on samples. So that's great. We can add a single guide. But we've heard from researchers that one guide is good, but if you could link 2 guides together, so make a double guide, that would be very useful, because now you can silence 2 regions on the genomes at the same time. And so you can now look at interactions between genes. And because we can make oligos of length up to 300 base pair, we can make those double guides very easily. The previous method of making those guides is extremely tedious, but now just go on a website, upload your sequence, and you're done. So that has been quite successful. And a number of more recent screening results from CRISPR screens have been published in the literature using the twist oligos. The next step, once you have the target, is to find an antibody against it. And so people use libraries of DNA to create libraries of either antibodies or libraries of proteins. And so the ability to create libraries of DNA mutant is very important. The problem is that the main tools of making those libraries are imprecise. Random mutagenesis only covers a small part of the possible changes. NNN synthesis, NNK synthesis, or trim synthesis have biases. They still have stop codons, so there are some issues. And so at Twist, because we can make a lot of DNA from scratch, basically in the computer, you tell us the mutant that you want, and we never make anything random. We can make exactly the mutant that you want. And so that means that instead of looking for a needle in a haystack, we can build you a stack of needle and make your work much more efficient. And that really relies on the silicon chip that we use for making our oligos. And that means that when we make protein libraries, when we make antibody libraries, you have base by base precision. You can control your codon usage. There's never anything random. We can then precisely control the diversity in that library. We can control the ratios of amino acids. We can control the length of either the CDRs or the length of the domains. We can remove unwanted motifs, or we can remove restriction sites, and we NGS sequence at the end. And so what that means is that you can have a very precise synthesis of your library. And when we compare again to NNN or trim synthesis, we have all of the benefit of full control of the amino acid distribution, no stop codons, no out-of-frame mutations, no liabilities, and very importantly on the right, we can match precisely the human repertoire. And so you know that 100% of the molecule in your libraries follow the rules of the human repertoire. And so those libraries are available to you. We also have at Twist a service. So if you don't want to discover the drug yourself, you can come to us and we can either discover antibodies for you. And we have a great track record of discovering antibodies for hard to drug targets. And we can also optimize antibodies for you and deliver biobetter. And we've done that many times. Here I'm showing a list of targets for which we have found agonist, antagonist, highly potent antibodies. Some of those are actually very hard to drug target. And so if you need an antibody, we can either have the tools for you to do the discovery, or we can do the discovery for you. And then in human health, last is around sequencing. Because the diagnostic of disease is very important to know exactly how to treat it. And we have a targeted sequencing solution, which enables you to choose how many regions do I really want to sequence to answer my question, and then what depths of coverage do I want. And so we enable germline sequencing, and so that's very useful for rare disease. We enable somatic detection, that is very useful for cancer detection. We even enable circulating tumor DNA sequencing, that is very useful for liquid biopsy, for finding at a very early stage if someone has cancer. And so, but if you don't want to sequence a big part of the genome, we can also enable minimal residual disease detection. So really there is a solution for whatever you want to do. And so the key to our targeted sequencing solution is that it enables a broad range of application, from liquid biopsy to rare disease, to oncology, to population genetics, to infectious disease, and more. But we do it in a way that is much more precise than what others companies has. For instance, on the right here, I'm showing you the blue square are the regions that we want to sequence, or the customer wants to sequence. And so what you want is sequencing signal that goes above the red line. So it goes up very sharply, goes down very sharply. So you sequence just what you want. And you can see with Twist, you have those very sharp peaks. And with other companies, you don't. You have those very broad peaks. Yeah, some peaks are high, some peaks are low. And so basically, it's not a good capture. And the reason why we are better than others, it comes from the synthesis and the amplification of the DNA. Because here I'm showing you on the left the synthesis of thousands of oligos. And when we sequence them, when we look as a function of GC ratio, the red line is flat. So that means that the synthesis is uniform for high or low GC. And then at the top, at which when we amplify the oligos, we found a way to keep that uniformity. That means that when we capture three regions, here for instance, the three exomes, the three regions, the coverage on those regions will be uniform because the oligos are uniform. However, if the amplification is not done well, you will have a GC bias. Some oligos amplify well, some don't. And what that means is that some regions will be over-sequenced, some regions will be under-sequenced. And so you're just going to have to sequence more to get your answer. And so the main benefit that we provide you is a lower cost per sample. And in sequencing, you have a fixed budget. And so if you will lower the cost per sample, you can do more experiments. And so here on the left, showing the price of our kit is about the same as the competition, but the cost of sequencing will be 50% less. And it's not only us saying it, the Broad Institute and many other customers have seen it. In addition, we can rapidly customize your panels, which means that you're going to get to your answer faster. And we have a fast hype, which means that you can go from DNA to the sequencer in one day. Finally, we've launched recently a methylation product line. And so not only you can detect SNPs and copy numbers and structural rearrangement, but now you can even detect the methylation state of your DNA. So to conclude on human health, we can go from target identification to target validation, to drug discovery, drug development, disease monitoring with the tools that we have. And then very briefly, we also want to improve the sustainability of data storage. And currently, a lot of the data is stored on tape and hard drive, and this is the Moonrise from 40 years ago. And when NASA went back to reread the data, reread the tape, you see that the tape is damaged. It's a very low quality image now. It's because magnetization is gone. And so with DNA, we have an opportunity to do better because DNA is permanent for millions of years. So the way data storage works in DNA, if you have a file, zeros and ones, you can convert that to a CGT. We can synthesize it, store it for 1 day, 1 week, 1 month, 1 year, 1,000 years. And then when you want your file back, you can retrieve it, sequence it, and decode back. We've done demonstration with Microsoft and Netflix, and it works great. So in conclusion, we are there for you. We have great products that hopefully will make your research easier. And if you have questions, if you have products that we don't yet have, please do let us know because we're constantly innovating, bringing new products out to make your life easier. So with that, I thank you very much, and I hope you enjoy this virtual event. Thank you.